So thanks a lot for the introduction and letting me present my work here today. Like Haley said, that I would love to meet you guys in person, but there's some issue with the visa, so I can't. But it's also nice to join you guys online today. So I'm Maggie Wong. I'm a molecular biologist working at the Max Planck Institute for Psycholinguistics. So after finishing my PhD at the University of Oxford, I joined the lab of Simon Fisher to study the genetics of language. So a little bit introduction of where we are located and who we are. So like I said, we are at the Max Planck Institute for Psycholinguistics, and I'm in the Language and Genetics Department led by Simon Fisher. We are located in Nijmegen in the Netherlands, very close to Germany. And this is our wet lab where we do all the experiments. And the results you're going to see a little bit in my talk today. So like I said, our department studies the genetics of speech and language, which means that we study how our DNA affects how well we can communicate to understand and produce speech and language and talk to each other. So, but why do we look at DNA and genetics? One thing you need to know is that we are all different from each other, and this difference is partly because of the genetic factors, because of the differences in our DNA. And what is DNA? Before we talk about DNA, we first need to know that our body are built with many, many different cells. And in each of the cells, we have DNA, which is a material that is existing in every single cell in our body. And it functions a bit like an instruction manual, and it has its own language, and it's written in a combination of 4 letters that you see here. And this DNA is very, very, very long. And in order to pack into one single cell, it has to wrap around and being very packed into this structure called chromosome. You don't have to remember any of those terms, but I have to go through a little bit of the background and the terms that you will hear quite a lot in the talk in order to follow it. But I'm going to not talk too much about the technical details, but there's few key terms that you need to know in order to follow. And so the next thing to know about is a gene. What is a gene? So SETBP1 is a gene, but what is that? So actually it is part of your DNA, and that have a certain pattern. And these genes, for example, SETBP1, some of them can give instruction to make a protein. And protein is something that inside a cell that can do its job in it. So that will control how well we function, things like that. And each of us had 2 copies of the genes. So each of us, for example, have 2 copies of SETBP1, and we get 1 copy from each of our parents. So we get 1 copy of SETBP1 from our mother and 1 copy of the SETBP1 from our father. And how do we get that? So we have an egg from our mother, we have a sperm from our father. And when they come together, they become 1 cell. And cells have to multiply, 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 and at some point to make many more cells to form our body. And during this process, DNA in our cells also have to copy itself, so that every single cell also has a copy of DNA. But during this process, it can sometimes randomly introduce some changes to the DNA sequence, and that's what we call DNA variation, or genetic variation. And this genetic variation can be very, very big, or it can be very, very small as well, only affecting a single letter. And it can be very common that you can find in a lot of people, or it can be very rare that it can only be found or even in a single person in the world. And these DNA changes can either be inherited, which means you get it from a parent, or it can randomly arise during the process of DNA copying itself, and this is what we call de novo. And most of the variants found in SETBP1 are de novo. So you don't find them it's not inherited from your parents. So, and all of us, each of us have many of these genetic variants. So we are all different. All right, okay. We covered the basics. Now we can move on to gradually talk about SETBP1. So, and today we're going to focus more on the rare variants that has very large effect that cause something called monogenic disorder, which means, for example, the very rare DNA changes in SETBP1 that can already cause some very obvious effects and cause, for example, haploinsufficiency disorder, and we can see clinical representations in the individuals with these SETBP1 changes. And how do we find these DNA changes? And with the new DNA sequencing method that you probably hear before already when you talk to your clinical geneticist or clinician or your GP or clinicians. So what we do usually is we group a number of individuals with developmental speech or language disorders, and then we screen their DNA. And then in that way, we can find the very rare DNA variants. And so far, what we've found, I want to go back one slide, yes. So far, what we found, the genes we found in these individuals are very important for early human brain development, including SETBP1. But the roles of SETBP1, among other genes, are still not very clear. And also, it is not completely understood how do the genetic variation I just mentioned can eventually cause, for example, SETBP1 haploinsufficiency disorder. And today I'm going to tell you how we can study that in the lab. Before that, we're going to go through a little bit about the genetic variants and what it actually means in your DNA and in your cell. Although Dr. Sid already briefly mentioned it already. So first we have our schizoid syndrome, SGS, which is caused by missense variants at the very critical region in the SETBP1 protein. And it's only affecting a very small portion of this region, which is very important for the cell to clear away the SETBP1 that is already used. And when there are these kind of variants located in this critical region, SETBP1 protein becomes more stable. So over time, there will be more SETBP1 protein in your body, in your cells. And these kind of variants cause, like Dr. Sid already mentioned, very severe clinical phenotypes in the individuals that have these variants. But if I can go to the next slide. Yes, but other kinds of SETBP1 variants, for example, that leads to, for example, SETBP1 haploinsufficiency gives a much milder clinical features compared to the SGS. And SETBP1 haploinsufficiency is caused by certain genetic variants, for example, deletion that is only removing SETBP1, or something that we call truncating variants, which means that the variants will make SETBP1 not, there will be less SETBP1 protein. So remember, we have, each of us have 2 copies of SETBP1, and these SETBP1 variants will only happen in 1 copy of SETBP1, so the other one is still functional. which means that in the end, we have less SETBP1 protein. And that's why it's called SETBP1 haploinsufficiency, also SETBP1-HD disorder. Go on to move to the next slide. Yes. So you already heard from Angela and Dr. Angela and Dr. Sid today about the very recent two studies that are led by Angela and also Dr. Brecher van Bon that we also contributed to. So from these two studies that we know that SETBP1-HD gives a very, very diverse disorder, and speech and language deficits is one of the very central feature in this disorder. But so far, it's still not clear what SETBP1 does during the early brain development, and also in the end, how do all the variants that I just mentioned can lead to the phenotypes that we see in these individuals, and why is it so diverse? And actually, we can study that in the lab, and I'm going to tell you how. So in order to do that, we make use of the latest development of cell culture methods by growing cells in the lab. So you're looking at a brain organoid. What does it mean? Some people also call it a mini brain. So what are they? It is a 3D structure that is composed of many different kinds of cells that you also see in the brain, and they are growing in an organized layer, which is very similar to what you also see in human fetal brain. That makes it a very good tool to study what's happening if there's less SETBP1 protein in the brain. And how do we grow this mini brain in the lab? So now we have to take one step back, because first we actually need cells. So we get the cells from people. It can be skin cells, it can be from cells from your urine, it can be cells from your blood, and we can magically make them into something called induced pluripotent stem cells, or just call it stem cells. And these cells are very magical. So if you give them the correct cues or hints, it can become different kind of cell types or tissue that you also find in your human body. And in our case, we want to make them into mini brain, or so-called brain organoids. So we have our iPS cells, and then we let them grow like a ball of cells. And gradually, this ball of cells will grow and grow bigger and bigger, and then the outer ring will expand and form these kind of pouches. That's something we also call a ventricle. But what is in these ventricles? What is in these pouches? If I can move to the next slide. Oops, too much. Yes. So if we cut the brain organoid open like this, and if we can use a method to label the representative features of these organoids, and you can see that actually each of it is one cell. You can see that these cells are organized in different layers. That is also very similar to what I just showed you in the human fetal brain. And in blue, you are looking at the precursor of a brain cell, and in the orange, you're looking at a more mature version of a brain cells, which means that we can use this model to study what happens if there's less SETBP1 protein in the system. Okay, so now we have a cellular model that can mimic the human early brain development event. How are we going to study the effect of the SETBP1 variants that we found? Again, we have the stem cells without the DNA change in SETBP1, and we call it a control. And we also have the cells that have the SETBP1 mutation in it, and that's what we call a mutant. And together, these cells, we would differentiate them or make them into mini brain, and we can compare the differences to find out what are caused by the disruption of SETBP1. And to get the control cells, like I said, we get from a donor, or we get from a parent that doesn't have the SETBP1 mutation. And how do we get the cells that is with SETBP1 mutation? If the slides want to show up, yes. There are two ways. One way is that we can actually make them in the lab. So we can make use of something called a molecular scissors. And that molecular scissors is going to cut your DNA. And your cells is going to try to repair your DNA. And during that process, if we only target SETBP1, then we can remove SETBP1 so it's not there anymore. And we can also get cells from the individuals that already have the SETBP1 mutation. And using this approach, we can look at what the SETBP1 do during early brain development, and also find out what are the pathways that are different compared to the control. And first, I'm going to talk about how we do that, make that in the lab. So again, using the gene editing method with the molecular scissor, we can make stem cells that has, remember I talked about the 2 copies of SETBP1. We can make it only missing 1 copy of it, which is, so you still have 1 functional copy of SETBP1, or we can make it missing both copies of SETBP1, so you don't have any SETBP1 in your body or in the cells. And we make them into organoids. And you can see that when it's in the control condition with both copies of SETBP1, it grows like a typical mini brain in the lab. But if there's less SETBP1 from the gross morphological, overall, it looks still kind of similar to when there are 2 copies of SETBP1, but inside there's a little bit of difference, but I'm not going to go into too much details in it today. But if you don't have any SETBP1, it looks completely different, and it shows a very, very striking differences if you just compare to the other 2 conditions. And at the moment, we are at the lab trying to find out what caused these differences. I'm trying to go to the next slide so that I can show you how. Sorry, the internet is a bit unstable. Yes, and so in this way, we can look at what causes the difference of the SETBP1. And like I said, we're also getting cells from the individuals that have already the SETBP1-HD disorder. And at the moment, we have made stem cells from the cells that we got from 3 different individuals and their sex-matched parents as control. And we're doing the exact procedure for these cells as well, like what I just showed you, so that we can understand what is going on and what are the pathways that are affected by these variants that we have here. If I can move to the next slide. Very sorry. It might happen in any second now. Oh. So that was the slide that I wanted to show you, but just skip it. If I can show you again. Anyway, so we talk about SETBP1-HD disorder, but I want to change a little bit of the focus to talk about, because we also know that apart from SGS and SETBP1-HD disorder, there's also, other missense variants that are not causing SGS, but they are a bit outside of the critical region that we just mentioned, leading to SGS. And in 2017, Rocio, who was one of the PhD students in our group before, identified that there are several, 3 of the missense variants outside of the critical region that are causing atypical SGS that you probably, quite a lot of you already know about. And over the years, we found actually there are more of these missense variants that are also outside of this critical region. And very recently in a medRxiv preprint that together with Raphael van Boon and also Angel Morgan, that we together look at the genotype and phenotype association, trying to link how these missense variants are causing the very big differences between the clinical features that we see, and also to find out which of these variants that we've so far seen are actually damaging or can be causing a disorder. So Raphael van Boon did the clinical phenotyping, and also Angel Morgan led the speech phenotyping, and I did the functional follow-up using cellular assays for some of the variants. And what we found in this study is that these variants actually cause a very variable developmental syndrome, both clinically and also in the cellular assays that we see. And in general, these individuals have lower speech and language ability, but all of these features only partially overlap with the classical SGS that caused by the variants within the critical region, and also only partially overlap with the SETBP1-HD disorder that I just talked about. And with the cellular assays, we've shown that there's a loss of function mechanism that is actually independent of the how much of the SETBP1 protein is in your cells or in your body. And we are at the beginning of understanding what is the spectrum of these SETBP1 variants, and also the related disorder that they causes. And so the study I just showed you was use a relatively high throughput way to screen a number of variants, but still we don't know exactly what are the downstream pathways that are affected and how is this changing the brain development. So then one of the next steps is that applying the same approach that I just explained to you, using mini brain or so-called brain organoid to study what are the effects of these missense variants that are outside that critical region. And by combining with the phenotyping of the clinical and the speech features, what we're trying to do is to try to first understand what are the roles of SETBP1 during the early brain development. And second is to find out if there's any common pathway or difference in the pathway that are involved in different kinds of SETBP1 related disorders. And how is it in the end giving very different phenotypic features that we see. And hopefully with that, we can improve or aid diagnosis so that we know better which kind of variants are causing which kind of disorders, and also try to use the information we get to inform better patient care. And hopefully in the end, we can identify some target that we can try to develop treatment for the SETBP1 related disorders. So with that, I'd like to thank the head of department, Simon Fisher, for letting me do all of this study, and also everyone that contributed to the project, and all our collaborators, and of course the BP1 Society for funding part of the studies that I just showed you today, and also of course for your participation. Without you, it will be impossible to do all this. So thanks a lot.